So my name is Helen Parkinson. I work at the EBI, and the title of the talk is Ontologizing the GWAS Catalog, a picture paints 1,000 traits. It's not quite 1,000, but it's very close now. So most of this work was done by two people in my team, Daniel and Antonio Baudet, who are talented bioinformaticians and programmers who basically started to deal with visualization problems for the catalog. So in this talk, I'll just run through a bit of introduction. I'll talk a bit about our infrastructure and our ontology. I'll talk a bit about what we do now to generate the GWAS diagram, and I will highlight some of the outlook from our technical perspective. So I guess you all know what the catalog is. I think the important note on this slide was that we were at a quarterly release of GWAS diagrams when my team started to collaborate with our colleagues at NHGRI, and the process was starting to fail to scale. And that basically means that manual extraction is very intensive and expert activity, and pulling the information that everybody uses in the catalog has to basically be done from the primary literature. And when you get tables that report more than about 5 SNPs, it can be a very intense process to get that stuff out, and really most of the data does come out of figures and items in the paper. So there were many, many papers. The SNP density on the arrays is going up, and more SNPs are being reported, and there are many more studies that come inside the inclusion criteria for the catalog. Okay, so we had a 2-year collaboration to take between my team at EBI and the vertebrate genomics team at the EBI, where some of the curation for the catalog lives. And really our aim was to start to look at the manual work and to try and start thinking about where the manual visualization versus automated visualization were. I want to go back one. And where we were looking at unstructured data versus structured data, and where we had essentially a very static visual interface, and start to think about how we could make that dynamic and responsive. And part of that's just a density problem. There's an awful lot of things that we would like to display on the diagram, and that's really a challenge to do that in real time and to have a diagram which really works. The diagram is incredibly iconic. When you go to a meeting, you see it in many, many talks, and that's one of the things about it that made it really important to preserve how it works, but also to make the process work of actually generating it, really available resource. So what we decided first to do was to examine the curation infrastructure. So my team basically examined the very labor-intensive and entirely manual process. And to support that activity, we developed an online tracking system, which automates a lot of the bits of the process, including automatically performing PubMed searches, entering papers into the system, helping with the triage of papers, assignment of papers, and also critically a thing called the SNP batch loader, which basically allows you to extract information out of papers and into the curation infrastructure very easily instead of having to type it. And this allows the productivity to go up and really means that the curators who are doing this work actually can employ their biological brains rather than their typing activity. They're all very highly qualified. It also allowed the curators to record the process and also to lead up to the quarterly release and organize the papers. And so this is just a little dynamic of what we first started to automate. So we started to automate the literature search, the data extraction, the data double-checking process, and finally the publication to the web. I should say that the genomic annotation part is done by our colleagues at the NCBI, by Mike Fiolio's group, and that information was automatically supplied to you by them and still is, and we're very grateful for that part of the work they do in collaboration with the catalogue and us. And then on to the second part of the work. So effectively we've now built an infrastructure for helping automate the data extraction, but where we initially came in and one of our more primary interests was looking at the catalog traits and starting to structure those in a slightly more organized way. So if you come to the catalog and you actually want to query it through the query interface, there's work here to do to structure this. So this is just a screenshot from the catalog at the point that we started working with it, where if you were interested in diabetes-related traits, they were available as a long alphabetical list, and they had different kinds of annotation on them. And these are basically what was annotated by the curators on the basis of the literature, but they're not particularly structured into a terminology. Initially, the curators working on this had started to use MESH, but MESH was not very well aligned for the sorts of data we had here, partly because we were often talking about things which really are phenotypes, so hair colour and eye colour, but also treatment responses. This is a very different kind of thing. There are a lot of drug response data inside the catalogue. Diseases themselves, type 2 diabetes, assays, for example, glycosylated hemoglobin, which might be an indicator assay or a diagnostic assay for a disease, and also chemical, drug, and protein names. Usually where it says things like C-reactive protein, it doesn't mean C-reactive protein, it means a measurement about C-reactive proteins. And then there are also some challenges about the way that the papers reported the data and the way that was extracted. So, for example, often the traits were compound and context dependent. So, for example, type 2 diabetes and gout, or Parkinson's interaction with caffeine. And in some cases, these generally are interaction traits, and it was kind of useful to have some way of combining these in an organized way so that it's possible to co-query them. We also know that there's kind of a long tail in the data, so that we see the same thing again and again. There are many more studies about diabetes and Parkinson's disease interaction with caffeine. And so we wanted to be able to reuse some of the annotation structures we already had to make sure that we could make those data readily available for query. So what we decided to do was generate an ontology for these data. So we basically integrated the traits with an existing ontology. We did consider creating a new GWAS ontology. However, my group actually manages data for various resources inside EBI. And the reason we wanted to integrate the GWAS traits with an existing ontology is we wanted to co-query data for genetic disease that comes from OMIM. We wanted to be able to query the trait information, and we also wanted to be able to query annotations on gene expression data, basically because we wanted to be able to interrogate the variation data with the gene expression data for reasons described in the previous two talks. And so really this allowed us to integrate all the semantic information into one resource and get to questions like, show me all the SNPs associated with type 2 diabetes and metabolic syndrome really quickly, and not just to do that query to return data to the user, but also to start to visualize that data using the same terminology. So my group delivers an ontology called the experimental factor ontology. It's already developed, and we have some development bandwidth for it at EBI. It's well suited to covering the diversity of GWAS traits. 20% of them were already found before we actually even started doing any integration of GWAS data. But what we did was we added around 500 new terms over 5 releases to give us 100% coverage of the GWAS data. There's a very high integration potential. It's used by other resources, including Ensemble and other databases inside EBI. But because we cross-reference lots of other terminologies, including SNOMED and ICD and NESH, actually it gives us an ability to have cross-referencing with lots of other data, for example, from the literature, but also from medical record if we go in that direction. So what we did was import the data from the GWAS Catalog. We combined it with our terminology, and we built some visualization software. And this process was done with the collaboration particularly of Kent Clem within the NHGRI, and also Lucia Hindorf, Heather Dunkins, and Peggy Hall, who actually generate much of the data that we were able to pull in and integrate. So I guess you're all familiar with the GWAS diagram. It provides a visualization of the trait associations. So the critical nature of the problem for me was that it's actually generated by a very talented graphic artist called Darren Laser, and you will see some of his work on the screen here. It was a static image in PDF or PowerPoint format, and because the catalog has grown so extensively, and I think a lot of people will have seen the animation of the traits being added over time, it had become a problem that there were too many traits and colors to reliably identify individual feature. It's a really great way for visualizing the evolution of the catalog. It was getting hard to interrogate the data and find all of the related traits and then visualize them against the genome. And so that's one of the problems we started to address once we had built a curation infrastructure and we actually had some back end that would allow us to deliver the data out to visualization. So this is where we came in. I think this was the last time that Daryl and Terry Manolio actually sat down to draw the data and visualize it manually. And this is the key at that time. And you can see that the key is quite large. There are a lot of colors. And also a lot of collapsing had already been done of over 800 traits by Terry just to make this key work. And it's kind of a computational challenge. So what we wanted to do was programmatically generate the diagram from our backend knowledge base. We wanted to have an interactive diagram that can be filtered by a number of criteria to show traits only associated in given scenarios. And I've written given disease here because before we started to do this, we analyzed the query logs. And we know that the top 10 queries for the GWAS Catalog are all disease-level queries. We wanted interactive traits, so dots that directly linked into the catalog, and a color scheme which could be used to visualize trait categories rather than individual traits, and that could be modified if needed to make the diagram look more visually appealing. So this is our new front end, and this is the diagram that we have generated. There's a color key which colors all of the dots into one of the hierarchies. There's nothing particularly special about this key. It was generated algorithmically. So we chose a certain number of colors, and then we mapped the traits into that number of colors based on the hierarchy and the ontology, so we would get a good visual representation of color. So there's nothing particularly complicated going on with the way the colors are arranged. We tried very, very hard to keep the color and the look and feel for the users, because the catalog is so iconic that we didn't want to turn people off by changing the way they interacted with it. And actually, the first version we produced, our colleagues at NHGRI told us it was too bubblegum pink. And I was like, okay, well, if the only thing you don't like is the pink, we can probably fix that. So that was a real achievement for us to be able to do this automatically. So the next thing we did was we started to put in trait-level querying. So you can now query the diagram by any single trait, combinations of traits, and also high-level categories. And this is the most common use now. We have generated little videos of how the traits change over time. We also have generated static diagrams for each time point of the trait, so we have it going backwards in time. And the reason for that is that the way that we have done the layout and the colors meant that the previous versions of the catalog were no longer interoperable with the current version. So dots had occasionally moved around in the past and color schemes have changed. And now the diagram is kind of backwards compatible. So if you look at the dot in the 2012 release, and you go back to, if it's in the 2005 one, you'll see that it's in the same place and it's the same color. The catalog is also dynamically linked now to various databases. So I've just shown a screenshot here of the kind of things we map to now. So you can get a map to the RSID for the SNP, you get the p-values that the curator has entered, you get our mapping term. We always retain the annotated terms that the curators annotate, but we provide a background mapping for those. So here you'll see an EFO mapping and the GWAS trait. The EFO mapping allows us to inherit lots of synonyms to improve the way that we provide queries to our users. The study is linked via PubMed, and the catalog is also linked. This is a screenshot from the catalog for the paper, so this will bring you into the right part of the catalog. But also we have now linked it to Ensemble. Ensemble uses the same terminology we do, so you can now start to query Ensemble variations with the same sort of terminology and use this terminology to query data from different sources other than the catalog. And Ensemble includes data from Cosmic and from OMIM and from other papers. From Ensemble, you can get out to the genomic context, the genes from regulation, population genetics, and LD blocks, and so on. And so this gives the user a jumping-off point to what could actually be quite a complicated landing point in a genome browser. So if I summarize where we've got to, we started out with an excessively manual visualization, and now we have an automated visualization. So we now are getting a data dump every month. We still release every quarter, but the bottleneck is no longer the visualization. The bottleneck is dealing with the volume of data that comes through the curation pipeline. We have highly structured data. We have a schema that describes both the data and the terminology. It's all built using semantic web technologies. So if anybody's interested, you can get all this data and you can get our visualization. And we had a very static visual interface, but now we have a fairly dynamic one. There are things we can do to make it more dynamic, and I'll talk about that at the end. We have put in the kinds of queries that we know people were doing against the existing GWAS catalog. We have done some surveying to decide what kind of things we would like to do next. I have put a quick techie summary in here. This is all built using fairly standard technologies. It's Java, it's running on Tomcat, it's an SVG diagram. It uses exactly the same diagram that the catalog was using before. There's a JavaScript library underlying it. The technically interesting bit for my group was that it's all built using an knowledge base, so it's semantic web technology. and it's all built using continuous integration. So it's a push-button activity. We get the data import, we push a button, and a visualization pops out the other end. All our code and data and schema, ontology, everything's available. We've already had some discussions with other groups. The carrier type display can easily be modified for other kinds of data, and some plant EQTL groups are interested in using it in other data contexts because they have the similar kind of display problem, although in their case they're displaying bars and not dots. But all of the infrastructure is fairly portable. So in summary, we've restructured the GWAS Catalog to allow querying. Before we were just doing direct string matching. Now we're doing tree queries. We are querying via synonyms, and we are doing hierarchical queries based on our mappings up to our terminology. This included going back and harmonizing all of the terms annotated for the entire catalog and remapping all of the catalog data to the terminology. This turned out to be necessary because the way things are done over time has changed. You don't do the same things back in the early 2000s that we're doing now. We have also modeled all of the traits explicitly, and this means we have teased out disease and measurement, and this allows us to be compatible with other projects in this space, for example, like Phenex. So it gives also some ability to be compatible with things like medical records in the future if we're able to be integrated with the way that people describe diagnostic assays. It gives us a little bit more power for the catalog's future use. We added a lot of new terms to the ontology to support the catalog. We didn't just add new terms to our own ontology. All of the response to drug or response to compound terms has been added at our request to the gene ontology, and then they're re-imported into our ontological space to do the queries. We've removed all the manual processing from the catalog visualization. So that's 3 weeks of time that took 2 people. That's basically gone, and it's automated. We're now supporting the curators to actually choose the terms during curation to try and make the annotation and curation more consistent. The literature isn't always consistent, but the curators are highly consistent. They have very highly prescribed processes. And we've used some semantic web technologies at our disposal to kind of achieve this fairly quickly. And the visualization actually took less time to build than the tracking system because it was fairly clear what the aim was, and the tracking process turned out to be fairly complicated, simply because extraction of data from the literature is actually a very complicated process. So in future work, we would like to explore some resolution strategies for high-density regions. The SNPs are not evenly distributed across the carrier type, and the MHC causes us particular problems. We don't want to redraw the diagram dynamically all the time. It's quite computationally expensive. With our colleagues at NHGRI, we're looking to capture, model, and query ethnicity information. So there are lots of challenges related to this. But actually, ethnicity information is something that in our survey of what users wanted, the respondents really wanted to see more ethnicity information. We had a kind of one-step-away integration from the genome browser, and that means you actually do have to go through a genome browser to see, for example, regulatory data. And we're interested in how we can bring that up more nicely and show elements of that in the context of the diagram in a way that doesn't land you into an environment where you have a very complicated genome browser layout. I had lots of things today that made me think there are certainly elements of regulatory data that we would like to display better. We want to be able to show per-study queries so that people can actually see if a study A compares with study B, so you could see all of the SNPs taken from a study at different time points. And also that would allow us to tease out things like meta-analyses and see sub-studies visualized against meta-analyses. So you could easily visualize what had changed in the meta-analysis. An important part of our work while we were remapping all of the annotation was that at the moment the papers are annotated per study and not per SNP. Although the catalog has always had the ability to store that information, it hasn't had the opportunity to visualize it. We've now made some modifications to our visualization strategy, and we will shortly be ready to provide a richer set of annotations because we'll be able to see SNP-level trait annotations. And this will solve some of those combinatorial questions that I highlighted in the early part of the talk, where you have multiple trait annotations collapsed because there are multiple traits associated with multiple SNPs in a paper.
How many minutes, please?
Sorry?
2 minutes, please.
Okay, thank you. And so the final thing is that we want to connect disease, phenotype, and assays. And what we think when people type things in is that they say type 2 diabetes, but what they really mean is give me everything you've got about diabetes, and that means all the assays, any related phenotypes, secondary complications, and also the disease annotation itself. And that's something that we're very close to being able to do. And I think I have one more slide on acknowledgements. Yeah, I do. So this is work which is funded by the NHGRI Catalog. We have lots of colleagues at NHGRI: Peggy Hall, Lucia Hindos, Heather Junkins, Kent Clem, Sarah Leah, and Terry Manolio. And several people in my group have contributed to the architecture of this. Most of the work was done by Tony Burdett, and Danny Welter, and Jackie MacArthur, and Danny are funded by the grant that's listed. Thank you very much.